speaker is Perry Marshall.
Good afternoon. My name is Perry Marshall, and that's my dad. I remember he was a pastor at a church in Lincoln, Nebraska when I was 16. It was a hot summer day. I was in the basement, and I heard him come in at 2 o'clock in the afternoon, which isn't normally when he would come in. And Mom says, "What are you doing here?" And he says, "I came back from the doctor, and the cancer is back." And she went to pieces, and she was an emotional wreck for the next 3 months. He died 15 months later, and I was 17. He was 44. The guy on the right is a longtime mentor of mine. He died of pancreatic cancer 11 years ago. The young boy there was a friend of my children. He died of leukemia at age 13. His name was Alex. How many of you got your own version of this story? Everybody. Well, I'm going to talk about how to detect cancer at stage -1. First, some conflict of interest disclosures. I'm the founder of a 501c3 called reversingcancer.org, and we are funding the projects that I'm talking about today. I'm also founder of a $10 million prize for the origin of the genetic code, which we announced at the Royal Society with Oxford professor Dennis Noble 5 years ago. 20 years ago, I went down the creation evolution rabbit hole. And I am an engineer by profession. I'm an entrepreneur. I'm an author. I've written books on Ethernet, Google ads, Facebook ads, 80/20, evolution. And when I went down the evolution rabbit hole, I'm just going to be blunt, I was appalled at the intellectual slop that passes for science in evolutionary biology. Somebody said, if you said, "Where does an eye come from?" and they said natural selection, that would be like, well, how did Kansas City win the Super Bowl? Natural selection. And what was their recruiting strategy? Natural selection. And what was their offensive strategy? Natural selection. Well, there's a great saying. Michael Eisen from UC Berkeley posted this on Twitter a few months ago. He said, "Every single thing people think happens in the smoke-filled rooms of science where power brokers collude actually happens and is way worse than you think it is." And I'm being a little inflammatory here because the cancer industry has been stuck for 50 years and it hasn't really made a lot of progress. And a person in stage 4 is really no better off now than they were in 1930. And what I have here is a chart of age-adjusted death rates for selected leading causes of death. And most of them are going down, and cancer is steady. And if you look up what is the definition of cancer, the NCI says it's a genetic disease caused by changes in genes that control the way cells grow and multiply. And I disagree with that definition. I think cancer is when the cells of your own body actively evolve changes for their own progress rather than for the collective. It's the cells of your own body responding to stress and evolving out of control. Now, the reason I'm talking to you about cancer is I wrote a book called Evolution 2.0, Breaking the Deadlock Between Darwin and Design, which I've talked about at previous ASA conferences. And I'm absolutely an advocate that evolution really happened. It's not a hoax. It's real, but it's also active. It's intelligent. It can happen very fast. And a major mechanism of evolution is symbiotic mergers between cells, such as mitochondria being a merger between a bacteria and a human cell. And cancer is the result of a symbiotic merger. And so let me walk you through it. Let's say that your kidneys are unhappy. They used to have a nice job. They used to like their department. They used to like their colleagues. They used to get breaks. Now the food is bad. The atmosphere is bad. The temperature's too hot. The temperature's too cold. The management is lousy, and they want to leave. And they they start switching on evolutionary mechanisms, including making copies of their genomes, going polyploid, and they want to leave, but they don't have any way to leave. And then a white blood cell comes along, and the kidney cell says, hey, if you give me a ride out of here, I'll give you immortality. And they do a symbiotic merger, and they merge together, and the white blood cell takes them swimming out into the bloodstream, and it now has an all-access pass to the human body because it's an immune cell, and that cell is a polyploid giant cancer cell. They appear in very, very, very small quantities in the bloodstream, and they eventually start spitting out cancer cells which attach to some organ in the body and metastasize. And that is where the metastasis of many cancers begins. I believe the number one person in the country on this subject is Jinsong Liu at MD Anderson. He says a supergiant cell provides a cellular platform to reset the genome for new organisms throughout evolution. I want to show you a little graphic of how this works. A stressed cell, number one, merges with stressed cell number two. They fuse. They form a polyploid giant cell. That cell starts producing tiny little cancer cells. You treat them with chemotherapy, it kills them. You did not kill the original cell. It can stay in your body for years, and then cancer recurs. The chemotherapy never takes out the original mother of the cancer cells. And so polyploid giant cells are major players at key points. And I want to skip forward and show you what I think is an absolutely brilliant observation. This is from Jinsong Liu's work. And on the left is the pathway of a normal embryonic cell when a baby is conceived. And on the right is the pathway of a tumor cell. And the paths are almost identical. The only difference is on the left is sperm and egg, and on the right is kidney cell and immune cell. And they do a merger, they fuse, they go through a set of cycles. Early in the cycle, the hybrid cell does massive genetic modifications in order to prepare its offspring for the environment that it's in. The tumor microenvironment is absolutely critical, and the organism is adjusting in real time to the environment that it's being born in, and it is building its defenses to ward off whatever comes. And a polyploid giant cell is a very tough animal. And so these are microscope photographs of the top is zygote to morula in embryonic development. In the bottom set of slides is polyploid giant cell turning into cancer cells. They're almost identical. And they are an essential component of relapse of cancer. Because again, chemotherapy doesn't take them out. And so if the giant cells are still in your body, eventually they're going to go back. There is a very similar type of cell called a cancer-associated macrophage-like cell, or camel cell, and they are found in the blood, and they have an extremely high correlation with stages of cancer. And so you can see the correlation between the percentage of patients showing CTC cells and incidence of cancer on the left, low incidence of CTC cells, low cancer incidence on the right, almost 84 to 97%. correlation. And the size of camel cells directly correlates to longevity numbers in cancer patients. So this indicates a much better way to detect cancer. And there's a machine made by a company in France called an ASA machine, which Azra Raza at Columbia University is using to detect the presence of polyploid giant cancer cells. And this is a way of detecting cancer at stage -1. It's like a glorified coffee filter. Now, I want you to imagine that there's a $25 test in Walgreens where you can go find out that whether or not you have polyploid giant cancer cells. And if you don't, then the likelihood of you getting cancer is extremely low. And if PGCC cells do show up, then it's likely that you're going to develop a cancer in the next 24 months. And you can do something about it now, and you can detect it early instead of the smallest amount of cancer that you can detect with CAT scans and PET scans is 1 millimeter, which is 100,000 cells. This is detecting it when there's only one giant cell in 7.5 milliliters of blood. So you're way ahead of the game. There's also a researcher at Columbia University that's developed a chip that can be implanted under the skin that will detect the presence of these cells, and then you can just monitor the chip and you don't even have to go to Walgreens. So imagine all of the people that could be saved if we were doing this. Well, so the problem is that the transition from non-cancer to cancer has never been studied in adequate detail. All of the money and resources are going to stage 3 and stage 4, where it is frankly too late to do anything about it. The horse is out of the barn. You already have 10,000 species of cancer cells, and the probability that any one therapy is going to take them out is nil. And that's why your sister-in-law called you and said, my numbers are so much better, and she was dead 4 months later. It's because when you start attacking cells that already have their evolutionary machinery switched on, 1,000 species of cell is going to turn into 100,000 species, and you're never going to catch up with it. The data already exists. It's just not being collected or shared. It's easy to deal with cancer at stage -1 or stage 0 or stage 1. It's very expensive and painful at the later stages, and early is 100 times more cost effective. So this is the ISET machine. This work is being done right now at Columbia University. Azra Raza is running the lab with her lab director, Abdullah Ali. And this slide shows you the comparison between a regular sized cell and a polyploid giant cell, and this machine detects these. And so if you want to study this, the best population to study is cancer patients who are already in remission. And so Columbia has started a tissue repository where every time remission cancer patients come in for their checkups, they take blood, saliva, stool, hair, and fingernail samples every 6 months. They log those. They monitor for giant cells. And when a patient develops a second cancer, they go back to that tissue bank and they do genomics, proteomics, metabolomics, all of the omics, and they thoroughly study it. And it's the first time anybody's had a prospective study to catch cancer at the very, very earliest stages. 20% of all new cancers occur in a cancer survivor. So since these giant cells are needles in a haystack, this is the best population to study. There will be 2 million new cancers this year. 400,000 will be in a cancer survivor. So what if we had access to a repository? So this is what we're building. So Azra Raza got 30 people together. They wrote an opinion piece in Scientific American in 2021 during COVID. And she got the members of that alliance to agree. These are people from Columbia, Harvard, MD Anderson, Johns Hopkins, Northwestern, and a few other schools. They agreed to take these samples. Columbia is already doing it, and we're fundraising for this. And it will be the first time that anybody has set up a study for stage -1 and stage 0 rather than the stage 3 and 4 stuff that's so common today. And so I am helping them do that. And these are my thanks and credits, and I am happy to take questions.
Great talk, by the way. I really enjoyed that. So actually, I spent a lot of part of my career as a cancer researcher, so that's kind of very exciting. I do know no matter how you detect it, you're always going to have a problem with chemotherapy. I was wondering if you could maybe comment, there's a lot of new treatments in the immuno-oncology space, checkpoint inhibitors, CAR-T, and bispecifics. Do you think that in combination with these early detection will make a difference, or maybe
yes,
expand upon it?
That's a great question. So one thing I neglected to mention is Azra's lab patented a bispecific antibody that only attaches to a hybrid cell. And so they haven't figured out how to kill it yet, but they know how to paint a red bullseye on it. And they're going through a list of ways to take that out, including CAR-T and some other things. So I believe that taking out that cell is at least some large step towards a cure for cancer. And then you have to ask the question, well, why were the kidneys stressed out in the first place? They can use single cell transcriptomics to figure out what organ the hybrid cell came from. So they can pin it down to it was lung, it was pancreas, it was bladder, whatever it is. Personally, speaking of my own opinion, I think chemotherapy is terrible.
Yeah.
It's awful. And I've had several friends who said, I don't think any of these doctors have ever had this done to them. And so personally, I am much more biased in favor of other approaches, like you said, including nutrition. Also, psychological health has a huge effect on cancer outcomes. Women with breast cancer that have high levels of support around them have 8 times better outcomes than women that report that they don't really have any friends. 8x. Being married has better effect on survival rates than chemo.
Very cool.
And that's an average marriage. So, yes, sir.
Thank you for your talk. When you get to be my age, you're concerned a little bit about the prostate. What would you comment about PSA readings, and do they at all give a window on camel cells?
I don't know. I doubt it.
And then what do they give? What does a PSA test tell you when the PSA levels rise?
I don't have enough familiarity with prostate and PSA tests to give you a good answer. I'm sorry. Yes.
Hello. Thank you for the talk. And I might actually be able to address that because I study cell biology in early prostate cancer. So I guess in general, PSA is unfortunately not an accurate representative of the prostate cancer progression. And it just shows like a protein that is highly expressed in the prostate when there's cancer. But even if it's like normal proliferation, it can still be expressed. So my question back to the talk is more about how do you define stage -1 cancer? Because when I see like when you are seeing like chemo-like cells, to me, it sounds like it's already pretty serious because normally you don't see these cells in like a healthy patient. So how do you define diagnosing at stage -1?
So polyploid giant cells do not rapidly proliferate the way that their progeny do. So they can sit there in the tissue for years. And this is partly why this has been ignored. This was first proposed in 1857 by Virchow, The first paper was in 1911, and this has resurfaced in the literature every 10 years ever since. And it's only been in the last 20 years that anybody's really gone after it and taken it seriously. So Jin-Song Liu tells me, he's a pathologist at MD Anderson, he could describe the physiology of 5,000 different tumors. He says, every tumor I've ever cut open had giant cells sitting there. And everybody say, well, ignore those. Those don't mean anything. So they don't behave the same way that regular tumors do. And so we call those stage -1. But when they start spitting out baby cells, like that picture I showed you, that would be stage 0. Okay. And then you're off to the races.
Thank you.
Any other questions?
Since we have time, just maybe to clarify, are you saying detecting those polyploid giant cells would be detecting cancer at an earlier stage than screening tests like mammograms, pap tests, etc.?
It's much earlier.
Right.
It's way earlier than that. So the challenge is making sure that you're not scaring people with false positives. Nobody likes that. And PGCC cells and camel cells do not always turn into cancer, but they always correlate with mortality. Sometimes it could be hepatitis, sometimes it could be stroke, sometimes it could be a heart attack, but they clearly show that tissues are stressed. I think we're still okay.
Very good time.
Go ahead. Yes, sir.
I don't know if you're familiar with the Michael Levin, the developmental biologist, but
good friend of mine. Great guy.
Okay, so you know.
Love his work.
Because he's talking about membrane potentials and how they vary between cancer cells and normal cells and the communication between the cells. So is that something that you're also looking at? I mean, the cells have to be alive to do that, but.
So I asked one of Michael's lab managers, it's not super well known that normal cells have a 0.07 volt potential. Is that 70 millivolt potential that is normal?
Stem cells are much lower.
And cancer cells have some other number.
It's much less.
And I asked her, do polyploid giants, do they have weird membrane potentials? And she said, I don't know. One of the things on the list of things to try at some point is using some of Michael's cellular electric field techniques to take out the cells. Michael's done incredibly. If anybody just go to YouTube and type Michael Levin and go get lost for 4 hours because his work is incredible. Yes, sir.
Thank you. I'll take the science as a given in terms of these giant polyploid cells. So I'm going to think about epidemiology for a second. It sounds like a screening approach is what you're proposing. So the question would be, a screening is really useful only to the extent that there's some specificity between the detectable preclinical marker, in this case the giant polyploid cell, and some degree of specificity between that and some outcome.
Yes.
Either a certain type of cancer or a certain organ cancer. So where is the research at with regard to that in terms of clinical utility? Because from the work I do as an epidemiologist, I'm concerned about billions of dollars and years of humans being subjected to the process you're describing, but the whole time we're refining that specificity in a way that could be harmful on a population level. So just throwing out, where are we at with that?
So where we're at is, this is why we need a tissue repository with thousands of patients, samples being, right now it's in the hundreds of patients. And it really needs to be thousands per year. And when you have enough patients going through this process with all of the analysis that I described, the answers to those questions will become very clear. Right now, we have a lot of small samples of anecdotal evidence that suggest that everything that I'm telling you is accurate, but you'll have a much more accurate picture after 10,000 patients. Thank you.
Thank you very much.